Welcome to another video of Immunology Simplified. Today I'm going to discuss immune response to viruses, which are obligate intracellular pathogens. Now here you can see that viruses, there are certain viruses present outside the epithelial cells. So if these viruses have entered the body through any portal of entry, for example the respiratory tract or the GI tract or even the skin, they encounter epithelial cells. And to gain access to these cells, first of all what they have to do is they have to specially attach to the receptors present on those cells. So if we think about two viruses, I'll just give the example of two viruses here. One is HIV, human immunodeficiency virus. It enters the CD4 cells or helper cells, and it can enter through CD4 receptor and the TCR or the co-stimulatory receptor, which is the CXCR4. On the other hand, if we think of COVID-19, COVID-19 can enter the epithelial cells with the help of a receptor called as ACE2. So here I'm going to just mention this: angiotensin converting enzyme 2. So this is the receptor, and it uses its spike proteins to bind to these receptors and then enter the cytoplasm of the cell. And the nuclear material is actually, you can see, the nuclear material is then seen inside the cytoplasm. Now it has to further move, this nucleic acid has to move and gain access into the nucleus. So here it will become part of the nucleus, and then there is mRNA formed, and these mRNA going to produce proteins, and again along with its nuclear material these virons are going to come out. So this is a very basic of how the viral infections, they enter the cell and then produce their own nuclear material and viral proteins, and then come out in huge numbers, and then this kind of viral replication can result in damage to the host cells. Now on the other hand, the host cells have got their own system or sensing receptors that are present. For example, the epithelial, all nucleated cells, they tend to have specialized receptors. One of the receptor I'll talk about is called as RIG1, retinoic acid inducible gene 1, and this can detect RNA, and beside that there is MDA5, melanoma differentiation factor 5, and both these can detect RNA viruses. The cytoplasm have got another type of receptor as well, and these receptors are called as CGAS, cyclic GMP-AMP synthase, and this is able to detect DNA viruses. So the nucleated cells, they have got a system of receptors, and these are like toll-like receptors, and they can detect RNA viruses and they can detect DNA viruses with the help of RIG1, MDA5 or CGAS, which can detect DNA. Once these receptors detect the nuclear material of a virus, they are going to activate. So there is intracellular signaling, and then it is going to activate special genes, for example ISG, interferon interferon stimulating gene, and these genes are then in turn going to produce mRNA, and certain inflammatory cytokines are going to be produced, and in this case in particular interferon type 1. So type 1 interferon can be alpha or beta, and these alpha or beta interferon are produced by the activation of either RIG1 or C gas. Once these interferons are produced, so this is the initial stage. Immediately when the viral infection attacks the host cells, and all nucleated cells have got these alarming systems, these receptors are going to be activated, and they are going to produce these interferon type 1 which are alpha and beta. Now they are going to perform very important function that is called as antiviral state. They are going to establish an antiviral state, and how they are going to do it: first, they can block the entry of these virons into the cell. So their entry is going to be blocked. They are going to perform a lot of functions. I'll mention some of them which they can do. Then they can also produce the exonucleases which can degrade these nucleic acids that are part of the viron. Also, they are able to block this mRNA synthesis of the viral mRNA synthesis. So they are going to prevent the entry of the viruses into the cell. They are going to block, or they are going to degrade the nucleic acids of the virus, and as well as they are going to stop the messenger RNA synthesis, and as well as the release of enveloped viruses. So if you see 1, 2, 3, and 4, all these are different functions that are performed by these interferon alpha and beta that are produced as a result of the RIG1, MDA, or C gas synthesis. They also have an ability, so they can first have an autocrine effect. So the cells that are producing it, they are going to act on themselves as well as on the neighboring cells. So the next cell which are on both sides of these epithelial cells, they will help them develop something which is called as antiviral state. So here is, I'm just going to draw the timeline here. So the first thing that immediately occurs is the interferon alpha and beta release, and that's the first thing, and it occurs immediately. And what it does is it creates an antiviral state. So it's trying to contain the virus. This first step of interferon alpha and beta release is trying to contain the spread of the viral infection and protecting the neighboring cells as well as the cells that are already infected with the virus. Now this interferon alpha and beta is not just released by the cells that are infected by it. In fact, there are immune cells, specialized immune cells that are termed as plasmacytoid dendritic cells, plasmacytoid dendritic cells. And these plasmacytoid dendritic cells, if you see, I have drawn certain receptors here. One receptor on its membrane is called as TLR3, and then there are two receptors inside. One is TLR7 and one is TLR9. So these receptors are specially designed to detect the viruses. TLR3, it can detect double-stranded RNA. The TLR7 can detect single-stranded RNA, and the TL9 can detect CpG of DNA viruses. Once they are detected, similar to the actions that take place here, intracellular signaling, and then these genes are activated and interferon alpha and beta are released. So I'll mention here that interferon alpha and interferon beta are released, and a thing you should remember is that around 50%, so around 50% of the interferon alpha and beta, type 1 interferon, during a viral response is produced by plasmacytoid dendritic cell. So they are very important cell, very specialized dendritic cells, and their job is to detect the presence of a virus, RNA or DNA, and then it's going to release interferon alpha and beta, and that will again initiate antiviral state against the initiation or establishment of a viral infection in the initial stages, and try to contain the virus. Now, the next step is the blood have got specialized T cells which deals with the viruses, and one of them I'm going to mention is NK cells. So NK cells, natural killer cells. They are part of the innate immune response, and they can detect the presence of viruses in host cells, and the different methods by which it detects is, one of the method is absence of MHC class 1. So all nucleated cells, they can express a receptor that is called as MHC class 1, which will express the protein of a virus, and as long as MHC class 1 is expressed on them, these natural killer cells, they are inhibited, they are not going to be activated. But some viruses, they kind of trick the genes, and they down-regulate the MHC class 1 expression, and here is when these natural killer cells, they will start detecting these cells which have got a virus, and then they will kill by two mechanisms. So one is the perforin and granzymes, so they can produce perforin which will punch holes in these host cells, and then granzymes are going to cause the apoptosis of these virus infected cells. Another system is Fas ligand is produced by these natural killer cell, and it's going to bind to the receptor on these epithelial cells, which is called as Fas receptor or death receptor, and once they bind to each other, it will result in again the apoptosis of these virally infected cells. So the second thing on the timeline is that these NK cells, they are there. So if you look less than 2 days, in less than 2 days, activated natural killer cells can be detected in people who are infected with viral infections. So first thing was interferon alpha and beta, which created the antiviral state. Second is the natural killer cells. In less than 2 days, you can find activated natural killer cells. Now the third cell that is going to act is macrophages. So there are tissue macrophages which are present in the tissues, and they are going to recognize as either free viruses. So if there are free viruses in the tissues, they will be recognized and they will phagocytose them, and at times they can also engulf these virally infected cells. So these macrophages, they will engulf the free viruses or the virus infected cells, and what they are going to do, they are going to start producing the pro-inflammatory cytokines like interleukin 1 and TNF alpha and IL-6. These are the pro-inflammatory cytokines that are produced by the macrophages. So after this, now this is happening somewhere around day 3, so 2 to 3 days the macrophages have participated, and the blood cells, T cells and B cells, that can go to the regional lymph nodes. So here is the regional lymph node, and imagine there are CD4 cells, CD8 cells, and B cells in the follicles. So CD4 cells have been activated by the dendritic cells. So here are the dendritic cells that have brought the viral antigen, and they have presented it to CD4 cells, and they have been activated. And same is true for the CD8 cells. They have been converted into cytotoxic T lymphocytes, and these CD4 cells are then going to help the B cells. So the follicular helper T cell, so T follicular helper will help the B cell, and these B cells will become activated B cells to produce specific antibodies against the viruses. Now all the activated cells, they have come here. So here is CD4 cells and activated cytotoxic T lymphocytes, and then we can also see B cells which have been activated along their antibody. So they will differentiate to plasma cells, and they will produce specific IgGs, for example, which will then perform their functions. So now the cytotoxic T lymphocyte, it's very important. It's the dominating cell that will help in elimination of the viral infection. So the interferon alpha or beta natural killer cell macrophages, they can do the initial job, but to eliminate the virus from the body is the job of cytotoxic T lymphocyte. They can eliminate established viral infections, and again they are going to interact with the MHC class 1, which is expressed on virally infected cells, and they also have got similar damaging mechanism like natural killer cell, that they are going to produce perforins and granzymes and induce apoptosis in these virally infected cells, or fast ligand method of initiating apoptosis in virally infected cells. So these cytotoxic T lymphocyte on the timeline are the next cells, CTLs, and it takes around 6 days for them to come and perform these actions of eliminating them. Now these activated CD4s, they are actually constantly activating the whole system. How? They can activate these CD8 cells by producing interferon gamma. They will activate these B cells further by producing interferon gamma, and they will also act on these macrophages, and these macrophages are in turn going to produce nitric oxide, because they will be optimally activated, and nitric oxide will be produced, as well as TNF alpha, tumor necrosis factor alpha. So this is the effect of CD4. It's kind of increasing the potential of all these cells, whether it's the cytotoxic T lymphocyte, it is the B lymphocytes, the macrophages, as well as even the natural killer cells. The natural killer cells will get the interferon gamma, and it will enhance the potential of these natural killer cells to further upregulate the receptors and find these host cells that have been infected by the virus, and continue destroying the viruses. And now there is a little bit more about these B cells. So the B cells which have been activated in the lymph nodes, and they have been differentiated into specialized against these viruses through somatic hypermutation and affinity maturation. Now these B cells are going to produce these antibodies which will coat the viruses. So opsonins, as an opsonin, they will coat them, and macrophages can engulf them. Another thing is the natural killer cell. They can kill the viruses or host cells by the process of ADCC. So there's a process that is called as ADCC, antibody dependent cell mediated cytotoxicity. And when the antibodies coat the viruses or the host cells, natural killer cell can recognize them and they can cause damage to the host cells. And macrophages, they will use these antibodies to engulf more and more viruses. Now there's another important antibody that I need to mention here, and that is IgA antibodies. IgA antibodies are normally seen at the basal surface of the epithelial cells, and these antibodies then enter the cytoplasm of the cell because they go through transcytosis and the secretory component is added, and then they will move further, and finally these antibodies are going to be released here on the apical surface. So this is the normal way IgA antibodies have to be released into the GI tract, in particular respiratory tract, and urogenital tract at the mucosal surfaces. Now, one thing that it can do is these IgA antibodies are very good at neutralizing. So, and they will interestingly do something which is called as intracellular neutralization. So they will neutralize the virus inside these epithelial cells on the way when they are crossing these cells to go to the other side. And the other specific antibodies, IgGs for example, that are produced against the virus, they are now going to perform the function of neutralization of the virus. And these antibodies, that's why, are very important against protection. So in the event of a repeated attack by the same virus, the neutralizing antibodies are very important because they neutralize these viruses and they don't allow their entry into the cell from where all the problem begin. And if the viral infection has now been resolved, the memory B cells of CD4, CD8 and B cells, they will be performed. So the one important thing that occurs at the end of viral infection is memory cell formation. So in the event whether it is CD4 cells, CD8 cells or B cells, their memory cells will be formed and they can generate a stronger immune response more quickly. And the last thing in this timeline that happens is the B cell formation, activated B cells against the virus, and that takes place after 7 days. So roughly around 7 days, or after 7 days, the antibody titer will start to increase and will reach the maximum level. So this is how the components of the innate and adaptive immune system take care of the viral infections. Now, sometimes during viral infections there are some pathological consequences that take place as well, and that is the result of 4 different mechanisms. One of them is called as cytokine storm. As I mentioned, when the macrophages detect the presence of a virus, or they engulf them, or they engulf the virally infected cells, they tend to produce important cytokines, the IL-1, IL-6 and TNF alpha, and these have then a lot of functions. They act on, for example, on the liver, and the liver then produces a lot of CRP in response to IL-1 and IL-6. They act on the brain and they act on the blood vessels, and they result in the increased permeability of the blood vessels. The clotting mechanism is activated, so there can be disseminated intravascular coagulation, and they can also result in circulatory collapse. So this whole thing occurs if there is increased viral load in the body. There is too much viruses. Persistent virus will trigger these macrophages, and there is a lot of cytokine produced, and this is called as cytokine storm. And this cytokine storm is produced in by viruses, for example SARS, severe acute respiratory syndrome, and they are also produced by viruses like H5N1, as well as by COVID-19. So these are the different viruses that are capable of causing a cytokine storm. Then another thing that happens is if the viral antigen persists, it can lead to T cell exhaustion, and this T cell exhaustion is actually caused by the persistence. So the virus is persisting in the body, and this will trigger both these T cells and NK cells to express special receptors, for example PD1 and NKG2. These are expressed by these cells, but they are the negative inhibitory signals. So these viruses induce T cells and NK cells expression of PD1 and NKG2A, and now these T cells will become less responsive to the infection. And this state is called as T cell exhaustion. It is caused by the expression of these cells, and viruses, they trigger the expression of these receptors, negative receptors, and as a result T cells and NK cells will be less responsive to the infection. And this is caused by different viruses like HIV and hepatitis B virus, as well as COVID-19. They are known along many viruses. These are the ones that can lead to T cell exhaustion. Then we come to another thing that is called as antibody dependent enhancement. Sometimes the antibodies that are produced, if they are poor or weak antibodies, these poor and weak antibodies, they won't help the immune cells to phagocytose and destroy these viruses. In fact, what will happen is that the immune cells that will take up these opsonized viruses, they will get infected. And this is called as antibody dependent enhancement. And this occurs in infections like dengue fever, viral infection, as well as in COVID. It's expected to happen. In fact, it's there are suggestions that perhaps it may occur in COVID-19 as well. And one last thing is molecular mimicry. Sometimes there is the similar shape of the antigen of a virus and body structure. So either a kidney or a heart, they may have some protein that is similar to the viral shape. And one of the example is Coxsackie virus induced myocarditis. So these are the pathological consequences of immune response generated against the viruses. And that's all. Thank you for watching this video.